show all window. There it is. All righty. Okay, there we go. Oh, it's my great pleasure to introduce today's speaker at our monthly STG meeting, Melissa Coleman from Scripps College in Claremont, California. And I have the honor of doing this because, of course, Melissa was my first graduate student. She joined my lab, my so-called lab at the time, because it was just me, in January of 1990, about 6 months after I started my first faculty position at University of Alabama at Birmingham. This was a pivotal event for me because not only did it double the number of people in my lab, but Melissa turned out to be an outstanding and tireless experimentalist who carried us seemingly effortlessly to where I wanted us to go, out of the basement of the building we were in, of course. Her first project in the lab, there we go. Her first project in the lab was to localize the projection neurons that innervate the STG by doing backfills of the stomatogastric nerve with biocytin, and then immunolabel each prep to determine which projection neurons immunolabeled for which peptide family members. This approach was successful, and it generated her first paper. And the results were foundational for us because it not only enabled the rest of her thesis work, but it also enabled much of the subsequent work my lab has done over the last 30 years. And as part of that follow-up to the anatomy paper, she identified these 4 projection neurons, which we continue to work with. As many of you know, Melissa went on to publish a set of lovely physiology papers identifying and characterizing these projection neurons and their influence on the STG circuits by using a combination of intrasomatic recordings and intraaxonal recordings near the entrance to the STG. But her primary focus was on MCN1, and from her title, I guess she'll give you a small flavor of that today. Parenthetically, some of you know the following story, but here it is for the rest of you. When we identified these projection neurons, whoops, sorry, there goes my mouse. When we identified these projection neurons, we needed to come up with a naming scheme. And so we decided that the most expedient way to go was to use a common acronym and then add a separate number to each one. And at the time, Melissa had already learned that the previously identified projection neuron in the esophageal ganglion, the modulatory proctolin neuron, was named on the basis of my own initials. And consequently, she thought it was most appropriate that we create an acronym that bequeathed all of the COG projection neurons to her. Consequently, we designed them to be called the modulatory commissural neurons with a number, or, of course, the Melissa Coleman neurons. Hence, MCN 1, 2, 3, 4, et cetera. Although, due to a cranky reviewer, MCN2 at some point became CPN2. About a year after Melissa, along with Dawn and Brian Norris, helped move our lab to Philadelphia, she defended her thesis and moved to Brandeis for her first postdoc, working with flies in the lab of Leslie Griffith. From there, she moved on to Duke for her second postdoc, working on the birdsong system with Rich Mooney. This system suited her really well, and she stuck with it when she set up her own lab at Scripps College in Claremont, California, where she continues to reside and train a generation of undergrads in the birdsong system, and where she is now a professor of neuroscience. In fact, I'm pleased to say that one of her former trainees, who actually just was visiting me in the lab a few minutes ago, will be completing her PhD in neuroscience here at Penn in about 2 weeks. So without any further fuss, let's learn what's new in the world of neuroscience and behavior in the world of birdsong from Melissa. It's all yours. Take it away.
You need to stop sharing.
And I have to stop sharing, which I have now done.
Okay, great. Well, Mikey stole some of my thunder. So I will, but I will continue and go ahead and tell my story because it actually, basically I'm going to tell you exactly what Mikey just told you. And I'll try to slim it down. And so since this is mostly I'm going to talk about my work with wrens, and that will be part 2. But I thought since this is an STG seminar, I was able to go back hundreds of years to my work on MCN1. And I actually had an undergraduate student in my lab yesterday, and I said, oh, look, this was published in 1990, whatever it was. And she's like, oh yeah, I wasn't born yet. She was born about 10 years later. So that made me feel a little bit old. So I'm going to tell the quick story that Mikey just told about MCN1 in the STG and show a couple of slides with that. So of course, when I joined the lab, as Mikey just said, I was his first graduate student, and turnaround is fair play here. And so here's Mikey in our lab in the basement. Notice that there's no computer.
I don't see your slides.
Oh, I'm not sharing my slides. Oh. The reveal. Thank you. Okay. Well, sorry about that. Now can you see my slides?
Yep.
Okay. All right. Okay. I'm going to talk about wrens, and I'm going to talk about, first I'm going to talk about crabs. All right. So.
Hey, there's my faculty meeting jacket.
I never knew when you wore that jacket. So here's Mikey in his office at UAB, and notice there's no computer on his desk. The computer is off to the side because of course we didn't use that many. And he's got his books over here. And then on the right is we actually went to visit where we used to get crabs at Neptune. And here's Mikey at the Neptune with the Neptune shirt, which I'm wearing today. So there we go. All right, so when I joined Mikey's lab, he'd just come from Brandeis working with Eve, and here they are at the MSMB course. And he had identified and characterized the effects of modulatory inputs to the STG, focusing by recording on their axons, the modulatory axons down near the STG. And so what we were interested in was characterizing, as he just told you, the inputs to the STG from the commissural ganglia. He'd already identified from the esophageal ganglia, MPNs, and see how those influence the STG. And so, as he just said, the first million experiments I did were to backfill the STN with biocytin and look at the cell bodies within the commissural ganglia. And then, of course, we wanted to know which way they went, if they went through ION or SON. And so we would cut either the ION and SON and identify those. And so just to summarize here, in one experiment, we backfilled with the STN with biocytin to label cells in the COG, but had the IONs blocked or cut. And so we found about 17 neurons in the COG that projected through the STN to get to the STG. And we now know 2 of those as MCN7 and CPN2. And Mikey lovely told you the story about how we got those acronyms. And will now throw Ron under the bus because Ron was the reviewer that told us to change MCN7 to CPN2. And so here's a nice picture of Ron and I way back in the day as well. All right, and then we also found that there were 2 neurons in the COG that projected through the ION to get to the STG, and we now know those as MCN1 and MCN7. And so I'm just going to focus on, because most of my thesis was on MCN1, I'm just going to show you a couple of slides at MCN1. And so MCN1 is SNAX1, and here's an experiment to show that. So I recorded simultaneously from the cell body in the COG and the axon in the STN, and then stimulated the axon here. So here's an action potential in the axon. Then you get an action potential followed in the STN, the ION. Can people see my cursor? Okay, great. And then in the soma. And then we can do it the other way where we stimulated it in the cell body and saw an action potential in the cell body, then an action potential in the ION, the STN, and then in the axon there's the STN axon. And I also wanted to show this because, of course, this was done prior to computers being attached to rigs. And so this was based on a Polaroid taken of an oscilloscope, which is fun. And then here's the picture Mikey just showed you of a fill of MCN1. All right, and what we found was that stimulation of MCN1 in the COG excited both the pyloric and the gastric mill rhythm, and that's shown here. So here I'm stimulating the MCN1 soma, and you get a back and forth of LG and DG. You elicit a gastric mill rhythm, and then you get an increase in the pyloric rhythm. So just as we saw with, stimulation of SNAX1. Now, how do you get this pattern? At the time, there wasn't a direct connection between LG and DG. I think that somehow that this is a little bit different now, but anyway, we got an alternation between LG and DG, but the circuitry didn't provide the mechanism to do that. But we knew from Mikey's work that LG inhibited MCN1's terminal in the STG, and that's shown here. So here we're recording from the soma, the axon, from LG intracellularly. And now when we stimulate the axon, we get action potentials in the axon. We also see them in the cell body. And then when we stimulate LG, you see that that inhibits LG in the STG. And then you don't get action potentials in the cell body either. Okay, so maybe this feedback within the STG was important for framing the activity that MCN1 produces to get the gastric mill rhythm. And you wouldn't necessarily see these inputs just from recording from this very electrically and physically distant cell body. So, and that's illustrated here. I'll just show two more quick slides to demonstrate that. So here, what we think is happening is MCN1 excites both LG and DG, but then LG inhibits the axon in the STG, the MCN1 axon. And so when it does that, DG does not fire. LG then fires because of its interaction with INT1, and also because of this electrical coupling. And so just here's to show the feedback from LG inhibits SNAX1, which reduces the excitation to DG. Very indirect path here. And so in the bottom here, when we're not exciting MCN1, we excite DG and then stimulate LG, there's no decrease in DG activity. But here, when we stimulate MCN1, that excites DG. It also excites LG. And now when we stimulate LG, there's a reduction in DG activity. And that's a reduction because we've inhibited MCN1 terminals in the STG. And so how we get that alternation is that MCN1 excites both LG and DG. When DG becomes excited first, and then LG becomes excited later because of its interaction with INT1. This inhibits the presynaptic terminals in STG. And so now when LG is active, DG is not active, and then the electrical coupling helps maintain LG's firing when LG fires. And that's just illustrated here on this experiment. And so here's an experiment we did where I just turned this upside down for ease. We recorded intracellularly from the left MCN1 axon, and which is down here, with an electrode that was filled with potassium acetate. And it turns out when you record from these axons with potassium acetate, they lose the ability to release neurotransmitter, neuromodulator. So you're only left with the electrical coupling. So now the MCN1 SNAX, this is all through electrical coupling. And here we stimulate the right COG, or right MCN1, and you get an alternation between LG and DG. So DG is active, and then INT1 inhibits LG, and then LG recovers from inhibition and fires. And then when we add electrical coupling to LG, you see that you get an enhancement of LG activity there. So, and so just to think about this for a minute, just because it was kind of fun the last couple of days, here, when we stimulated MCN1 in the commissural ganglia, that excites the pyloric and gastric mill network. But how it does that, the mechanism of that, is not apparent from recording from the cell body, and is only understood when you understand all of the interactions that are occurring at its very electrically distant terminals. Okay, And so that is my, oh, I did pretty well. That's my quick 10-minute tour that Mikey gave a very nice introduction for of some of my, highlighting some of my work in his laboratory. Any questions about that before I go on to other things? Excellent. Okay, so what I'm going to talk about next for the remainder of the time is talking about work I started with Eric Fortune, who's maybe on this in this presentation, and looking at how basically duetting in plain-tailed wrens that live in Ecuador. And this work was really informed by my STG background. And thinking about pattern generating circuits and how you get a pattern and how that pattern is modulated or changed. And so I'll sort of highlight that throughout the talk today. And so what we think of is having two animals that are interacting, and each animal has a pattern generating circuit in its head. And how they duet is an interaction between those two pattern generating circuits. All right, and so and so here are the wrens that we record from in Ecuador. These are called plain-tailed wrens. You can see why they're plain-tailed wrens. On the top is the female, and you can tell that because she's got a pink band on, and then the bottom is the male. And they sing these lovely duets. Okay, and so these are experiments with Eric Fortune here on the left. Here's me again, and then Nancy Day, who's at Whitman College in Walla Walla, Washington. All right, and so these questions that we're addressing is sort of globally, how does the nervous system produce social behaviors? And that is an interaction between at least two individuals, maybe more. And so I've just highlighted a couple of the classics here. So here you have the blue-footed booby, where the male does a little dance to show off his blue feet. And so he's the sender in this relationship, and the female is the receiver and is receiving that information, and then is going to choose to see whether or not he did a good job. Of course, when she decides that, she will now become a sender and send information to the male, who might then change his behavior and become the receiver, because he's now receiving information. And you can do that with other species as well. So I've got here, birds doing the little waggle dance. They're receiving that message, but then they're going to become a sender and send the message to someone else. And then you might also have here some aggression in fish. So you have a sender and a receiver, and the receiver then will change its behavior and send a signal back to the sender. And so this relationship between sender and receiver is pretty complicated and can change over time. And one of the great examples of senders and receivers is in turn taking, in vocal communication. And so really what we're looking at is what's the neural basis of cooperative behaviors like turn taking, and that's illustrated here. And so whenever you converse with someone, you are going to take turns with that person when you say something. And so here the person on the left is saying something, and she is the sender. And then that message is being received by the person on the right. And then, of course, for effective communication, the person on the right then will respond, and she will become the sender, and the person on the left will become the receiver. And so it's this lovely back and forth turn taking between those two individuals. So how does the nervous system do that? So we're studying this in the plain-tailed wrens, I just showed you, these duetting wrens. And first what I'm going to do is just play you a video of the duets. And so on the top is the male, and on the bottom is the female. And then running across the bottom is the spectrogram of the songs. So it's amplitude over time. And then we've color-coded that so that the female parts are in pink and the male parts are in blue, just to keep things simple. So I'll play that now. And people could hear the sound? Yep, okay, great. And I'll play it again just so you can see, but if you close your ears or if you close your eyes, it sounds like a single bird is singing, but it's actually this really quick alternation between the males and the female. And just because it's kind of pretty. So it's a really remarkable behavior. So what I'll tell you about is what we understand about how the behavior and how the behavior of this, and then how the nervous system produces this. So all these experiments I'll be telling you about were done in Ecuador. These birds live in the slopes of the Andes here on the right. So here's Ecuador, part of South America, of course. And the Andes run this way, and they're located on both the east and west side of the Andes. And our field site is kind of here where my cursor is. And then here's Eric and one of our Ecuadorian colleagues, Carlos, who's now a postdoc in New York, looking for our birds in this very thick bamboo where they live at about 2,200 meters above sea level. And here's where we do our work. So this is one of our field stations, the Yanayacu Biological Field Station. We do our experiments in this corner room and sometimes house birds up here. We also live in this facility. And we have to use machetes to go through the bamboo to set up our nets so we can catch the birds. And then it's actually a very, very beautiful place to live. Okay, so back to the behavior. We started these experiments based on this paper published by Nigel Mann in 2006. And this actually started at NSNB. And here's their recording of a duet. And on the y-axis is frequency. So a low frequency would be down here, and then a high frequency will be up here, this is in kilohertz, over time. And what this shows is that the duet has a repeated unit, ABCD, 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 and that is an alternation between the male and the female. Did someone ask something? Okay. That's an alternation between the male and the female. So here the male, this is an up frequency, whoop, and then comes down, whoop. I'm not very good at that. And so you can see that where here the male sings and the female sings and the male sings and the female sings. And I can play that for you. Okay, so how do they do that? And I've got my talk into two different parts. One is the behavior, and the second is the neural activity. So first, what I'll do is we have two birds. One's the sender, and one's the receiver, excuse me. And then, so here the male will send a signal vocalization to the female, who's the receiver, but then the female becomes the sender and sends a signal to the male, and he now becomes the receiver. And so what I'll talk about is what are these signals that are sent between the two animals. And in fact, it looks like it's the auditory signal that's important, not surprisingly. And then talk about what's happening in the brain of the sender and what's happening in the brain of the receiver. What does that look like? Okay, so first, what is that signal between the two individuals? And what I'll show you a couple of examples that it's actually an auditory signal sent between one and the other. And the first example of that came from just recordings we made from the field. So I went out in the field and placed microphones in the field, and we just recorded song whenever they sang. And here's an example of this. Here is a duet. And we labeled, again, the female syllables in pink. So she's singing ABD, ABD, ABD. And the male singing C and E, C and E, C and E. And then on the top is the transition between the female syllables. So here, this is the transition time between syllable A and B is in green, between syllable B and D is in orange, and between syllable D and A is in yellow. Okay, and I'll play that song for you, this duet. All right. And then sometimes both the male and the female sing solitary song. So she sings in the absence of a male joining in the duet. And what we noticed is that when she sings solitary song, the sequence of her song is the same. So she's still singing ABD, 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 but the timing in between those syllables has changed. And that's illustrated here. So for example, the timing between syllable A and B has lengthened. We've got this now longer dotty green line. And the timing between syllable D and A has shortened. And we see the same thing in males when they sing solo syllables. So that's illustrated here. Oh, I forgot to play that one for you. You can actually hear the, well, at least I can. You can hear the gaps produced when there's only a single bird singing. Okay, and so you see the same thing also in males. So here is
Melissa.
Yes.
I'm sorry, I can't hear. Is this me or I couldn't hear any song?
No, I can't hear him either.
Oh, maybe I'm okay. It was working earlier. I'm going to stop sharing and try share again. I've got sound on. Can you hear that?
Very slightly.
I can't.
Isn't there a setting to share your audio?
Yeah, I thought it.
Yes, under the three dots, when you click on the three dots.
Yeah, I've got it sharing.
Oh, it's already on.
Maybe I won't optimize it for video clip. Sharing. What else can I do? Oh, wait a minute. Maybe if I do. What if it's coming out the microphone?
Melissa, mute your own sound, your computers, not your, and play it now, because it may be canceling the echo. No, it doesn't work.
Well, that's unfortunate. Audio settings. Hear that? It was working earlier because we tested it earlier. Huh.
Can you sing it?
The example had sound, video. The video had sound. Oh. Let's try one more thing. I'm going to close it and reopen it. Sorry, Farzan, what did you say?
I asked you to sing it.
Oh. All right, let's try that one more time. Share sound. Anything? Oh, I don't know what else to do. Sorry. Well, darn. Okay, well, maybe hopefully it's on just these for some reason. I won't play those again. Hopefully the video will play. That's the more important one. Okay. All right, well, I'll move on. Sorry about that. I'm not quite sure what's wrong. All right, so we were looking at kind of making a case that it's auditory feedback between the two animals that's involved in the timing. And that's also shown for the male. So here's a duet that I will no longer play, where the female singing AC, ACE, 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 and the male is singing BD, BD, BD. And then later, the male sang by himself, solo song. He's still singing BD, BD, but the timing in between those syllables has changed. So now the timing between D and B has decreased, and the timing between B and D has increased. Interestingly, when the male sings solo songs, the amplitude of those syllables is much, much reduced. And I can say maybe why that's the case. We don't really know. All right, and this change modulation of the timing actually occurs on a very short time scale, and that's shown here. So here's a duet that occurs, and at this point the male drops out. When the male drops out, the timing of the female syllables changes in just this 1 or 2 seconds. And so here you have the female singing ACDE, ACDE. And then when the male drops out, this timing between A and C increases. So this happens on a really quick time scale. All right, and so how we interpret this result is that there's actually a pattern generating circuit in the brain of each individual that's producing that pattern, ABD, 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 but it's sensory input from the partner that alters the patterning or the timing of that pattern within the receiver. All right, so what is that external signal? I'll give you two kind of fun experiments we did to test this to show that it seems to be auditory. And the first experiment is showing that visual input is not necessary. They don't like it, but it's not necessary. And so hopefully you'll actually will hear this. This is a video of 2 animals that we've caught. The females here on the right that you can see, the male is in the cage on the left that you can't see. And we prevented them from seeing each other by doing this very complicated experiment where we put an umbrella in between them. And so here I'll play that. Could you hear that? Yay, okay. So the videos work, so that's the most important part. All right, so what that shows you is that they can actually duet perfectly well without seeing each other. And in fact, if I went back to that first experiment where I showed the duetting, they weren't really looking at each other. But that's a nice example of that. And then to show that altering auditory timing can alter the duets, we did the following experiment in which we placed the birds some distance apart and actually changed the distance. And so, and that's shown here. So here we have the female at the top with a microphone near the female, and the male at the bottom with a microphone near the bottom. So here's the male in his cage, and there's a little microphone next to him. And then we increase the distance. And in fact, if it's auditory signals going between the two, then what we should see is the intersyllable interval should change related to the distance between the 2 birds. And that's the experiment I'll show you next. So we did this up to 7 or 10 meters, and that distance was limited by basically the size of the porch that we were on here. And sometimes we also removed so they couldn't see each other. And so here's the experiment. Here's the audio that was picked up on the microphone next to the female. And on the bottom is the audio, the duet that was picked up on the microphone next to the male. And one of the things this experiment helped us do is also figure out who was singing what. It can be pretty complicated, but of course the microphone next to the female, those syllables, the female syllables will be louder. And so here you know that the female is singing these syllables because those are louder, they're more black. And on the bottom, you know that the male is singing these syllables because those syllables are darker. And so that's also very good. So here, when the female sings then, you see her syllables on the microphone next to the female. And then after some delay, that's recorded on the microphone next to the male. Then the male hears that and responds. So if this auditory feedback is not is not critical, then moving the animals further apart or closer together, you actually might get some overlap. But if they are using those auditory signals to modulate the timing between the two, then you might expect that intersyllable interval to increase with distance. And in fact, that's what happens. Oh, sorry. And so I'll just move through that. And in fact, that's what happens. So here is distance, the distance between the two birds. And here, this is the intersyllable interval for the male and for the female. And what we find is that that intersyllable increases over distance and in a range that's what you would expect for the speed of sound at that level. So those two experiments really tell us that it's the auditory cues between the individuals that are the critical signal from the sender to the receiver. So now we can look at this a little bit more and say that each bird has a pattern, seems to have a pattern generating circuit in it, but it's auditory feedback from one animal to the other animal that changes the timing of when they produce a syllable. All right, and so how is the nervous system doing that? How is the nervous system producing the response, and how is the nervous system responding to that response? And so we then wanted to record from the brain as the two animals were interacting. And we just, where do you record from the brain? And we decided to record from this area called HBC. This is a cross section of a bird brain. This is a sagittal section. So if the bird faces this way and you cut the face that way, well, actually this way, it's through the midline basically. And HBC sits right up here on the top. It's an aggregation of thousands of neurons. Some of those neurons project through the motor system and eventually innervate muscles that control singing. So it's premotor. So HBC is premotor, which is important. HBC, we know from recordings in the other bird I studied, little zebra finches, that this HBC also can change the pattern generating circuit for song. So that also seems like a great place to be. Neurons in HBC also receive auditory feedback. And so here, that's kind of illustrated here, where you have auditory information comes into HBC, that then projects out, and HBC is also part of the motor pathway. All right. So what we predicted is that there would be premotor activity in HBC because HBC neurons code for the temporal features of song. It's the pattern generating circuit, so the timekeeper. And that we would also see auditory responses in HBC to the partner song, because as I've shown you, we have to have auditory input to synchronize the timing between the two verbs. All right, so those were our predictions. And so we went to go do it. Here's our early our early laboratory we set up. Here, it was here on a dirt floor with no windows or doors. Here's me with our microscope and our electronic equipment. If you'll notice on the left, we have a little thing to keep bugs from flying into our experiment in the evening or really anytime. And then here on the right is our ground, which is a copper pipe we had to put into the ground, which was super fun. And then later we upgraded and moved inside. And so here's us setting up our laboratory inside. Our table, of course, is a door that we got from down the road. And the legs of the table are suitcases we had. And now we're setting up a microscope and a heating blanket and some anesthesia machines and our extracellular devices, etc. And then what we did to record from HBC while the birds were singing is we implanted extracellular electrodes in HBC of each individual. And then that signal was amplified and digitized on the head of the bird. And that's shown here. So here's a bird with this, the little blue thing is the amplifier and digitizer that we put on the bird. It's wireless. And so that then sends a signal to a receiver that's also in the cage, and that then is taken to a computer. So that's the bird with the that we recorded from the HBC. And then here is Nancy making those electrodes in the field. Okay, So what I'll show you two videos. They're actually the same video, just one of them has neural recordings on it. And so here's a video of the two birds duetting when they each have electrodes in HVC. Okay, and you could hear that now? Now I'm like totally paranoid. Okay, great. Yeah, and so there they are with their little backpacks on. They also have to have a battery, of course, to charge that. So their little backpacks on and they're duetting. Now, in these animals, because we had electrodes in HVC, what I'll do now is play for you a version where you can hear HVC activity in the male and the female through little tips. There we go. So lower pips are the female action potential and higher pitched pips are the male action potentials. And first we'll go through and you'll hear both of them, and then you'll hear only the male and then only the female. Oops, sorry.
Melissa, your control bar is in the middle of the data.
Thank you. I didn't know what you could see. It's really annoying. I'll go back. I'll just replay it.
Hey, Melissa.
Yes.
A quick question. Is this, are these electrodes picking up all of HVC activity? Does it matter where in HVC the electrode is placed to get this?
We basically kind of did the hammer over the head experiment where we're just trying to get a pole into HVC. So there's probably, it's not all of HVC activity. And so there's fewer neurons than all. I don't know what the numbers are, maybe like 20 per recording site. And in fact, as I'll show you next, we have up to 4 electrodes in HVC. So we can kind of sample across that. We haven't done a dive to know if it matters where you are. From recording from other birds, it doesn't seem to matter. But we haven't gotten to that level of precision yet.
But each time you put an electrode in HVC, you can get this kind of rhythmic activity.
Yes.
Cool.
Yeah. Okay. Sorry, I'll try and move that thing out of the way. I didn't realize it was there. All right, so we made some predictions that there would be premotor activity in HVC. And because HVC neurons code for those temporal features of song. And so what I'm going to do is walk you through that experiment that I just showed you, but in graph form. And so here is the spectrogram of the duet, where we've labeled the female syllables in pink, just to be, or at least the timing, just to be easy. And so here the female singing A, B, D, A, B, D, A, B, and the male is singing C, E, C, E, C, E. And we have electrodes in HVC of each one. And so that's shown here. So again, we have up to 4 electrodes in HVC, depending on how lucky we got. And so I'm showing you action potentials from the electrodes in those 4 electrodes. And so the male action potentials are on the top in blue, and the female are in red and pink, really. And so here's the activity, wake and singing. And then, of course, we can look at those rasters by looking at a histogram. And so that's shown here. And the histogram in the male, we've just put upside down so you can see the alternation of activity between the male and the female. And so here, when the male sings C and E, you see that there's an increased activity in HVC of the male. And when the female sings, there's an increased activity in female HVC. And you get this nice alternation between male and female behavior and the brain activity of HVC in the male and in the female. Okay, and so our prediction was that you should see an increased activity when in HVC when the bird sang. And in fact, that's what we see. And we analyze that just looking at response strength. So how much activity increased in HVC compared to baseline when each animal was singing. And so this is the response in the female. You see an increased activity in HVC when the female sings, and an increased activity in the male HVC when the male sings. So in fact, there is increase in premotor activity in the sender as we predicted. So yeah, we got that right. Okay, so our other prediction is that there would be an auditory response in HVC to the partner song, because of course I showed you that lovely behavior where they're really dependent on auditory signals from the partner. And so they need that to coordinate the duet. And so here's the same experiment I just showed you, but now what we'll do is look at the other part of the duet, and that is when they're receiving that information. So here, for example, the female singing, so you see an increased activity in HVC when the female sings, but you see really nothing in the male when he's hearing her song. And the same for the male. When the male singing, you see an increased activity in HVC when he sings, as we predicted, but there's really nothing in the female. And this was really confusing to us because we knew that auditory information was so critical. We didn't quite know what to do. But then I have that amplified here so you can actually see there's just not much there. But then we went and actually measured. And when we measured, we found that there was a decrease in activity in the brain HVC of the receiver compared to baseline level. And this decrease in activity then suggested to us that HVC was inhibited when it was receiving information from the partner. So we predicted that there would be auditory responses in HVC to the partner song, and we predicted they would be excitatory, but in fact, so we were correct with that, but kind of with the wrong sign. So we think that there are auditory responses, but they're actually inhibitory. And it's what we'd like to do next is actually show that. We did one experiment I'll show you next that sort of indirectly looks at the inhibition between those from one HVC to the other HVC. And in the same
Melissa, is it feasible to stimulate the HVC at random phases to construct a phased response curve?
Yeah, so we'd really like to do that experiment. That's like, you know, the list of experiments we want to do. And of course, this builds from the STG work. And now the recording setup we had in the birds I showed you earlier, they now have a way to stimulate. And so the next experiments we want to do is go back and do that exact same thing. So as they stimulate in HVC, and what we predict is that it'll cause some phase shift. We know people have done that in finches several years ago, and you either get a reset, so the birds start at the beginning of the song again, or they completely stop. So we should see something kind of fun. Yeah. Great idea. Anything else about that? Thanks. That was a great question. Okay, so we did do this one experiment to test for inhibition, at least just initially to do that. And that at the end of this experiment, after the birds sing, we then anesthetize them with this drug called urethane. And urethane does a bunch of different things, but one of the things it does is it blocks GABAergic inhibition. And so when we did that experiment, what we found was we uncovered auditory activity in the brain of the receiver. And so at the top, this is a little overwhelming, but at the top is the experiment that I just showed you. So here they sang the song, and you see this nice alternation of activity between the male and the female. Now, when we take those same two birds and we anesthetize them with urethane, and then we actually changed electrodes, and then put an electrode back into HVC. Now when we play the same duet back to those individuals, so now they're only listening to the song, we find that there's now an auditory response in the brain of the male, for example, when he's listening to the female. So now you see these increase in activity in the male when he's listening to the female. And the same, but lesser so in the female, when she's listening to the male, there's now an increased activity in her brain when she's listening to the male. And so, and that we also quantified here, where you see an increase in the brain of the receiver in both the male and the female. Okay. So what we think now that we have is that you have a pattern generating circuit in the brain of each individual. When the male sings, that comes from this pattern generating circuit, CE, and the female receives that, and her brain is inhibited, and so she doesn't sing over the male. Then, and that alters the signal in her brain, so then when she sings appropriately at the appropriate time, sends the signal to the male, and his brain is inhibited at that time. And this idea of inhibition between the two individuals is kind of interesting because for a couple of reasons that I'm sure you all have thought of. One is that it keeps the two birds from basically talking over each other. So when one is, which is important for turn taking. So when one is active, the brain of the other is inhibited, and it prevents that bird from singing, and therefore coordinates this turn taking. The other thing that's kind of interesting about this is, as you saw, the interaction between the two is very, very quick, right? So one sings and the other sings very quickly. And perhaps, we have no evidence for this, but I like to think about it, perhaps this is due to post-inhibitory rebound. And so when you get an inhibitory signal in that motor activity, it can respond very quickly and send a signal out, very quickly. So we're kind of excited about this, and our next set of experiments are really to more directly show that it's inhibition that's really sculpting this turn taking. And I guess I was nervous and went too fast, but and so I just that's all I have for today. But I wanted to thank the birds for their cooperation, so to speak, and then I had a bunch of students working on that myself. This was done at the Yanayaku Biological Research Station. We received some nice funding from the National Science Foundation. And then we had help from people in Ecuador. And we used this guy who's at Virginia Tech. We used his truck because, of course, we had a truckload of equipment that we used. And with that, I also went through boxes of photos and found other photos that I will leave for your amusement as we take questions.
And Melissa, quick question.
Yeah.
For this feedback, when I think of like rebound, like you just mentioned, I think of like a strong inhibition. And so I was wondering if you have ever tried like different volumes of feedback, like if when you had like the birds in the cages and you use the microphones and the loudspeakers, did you change the volume of the feedback and see if at some point this interaction breaks down, or if it's a graded breakdown or all or nothing thing?
Yeah, we don't know the answer to that. So for playbacks, it's interesting. In finches, volume doesn't seem to matter that much. With these birds, we've tested a little bit. It doesn't seem to matter that much, at least when they're anesthetized. Clearly, there's a change in behaviorally, volume's going to matter, right? The male activity is much reduced. The volume of the male solo song is much reduced. So behaviorally, that means something. So we haven't tried. That's an interesting idea. So the idea would be you get more volume, so you get more inhibition. So maybe you get more.
Yeah, or it's maintained over a certain volume range, and then it breaks down at one point, and it doesn't work anymore.
Right. That's hard to do because you have to get the birds to change the volume. The other complication with that is if their birds are awake and we play back song, they don't respond to that. So they don't call back to that. And also we don't see a response in HVC when they're awake either. So there's a couple, it's an interesting idea. I don't know quite how to do that. The other one that we thought about doing is kind of a la Eve's paper with, increasing the temperature. What if you just increase the distance so much that kind of the pattern generating circuit falls apart that way? And so we would like to go back and try those experiments. Of course, that's complicated because then you have birds in a cage with distance, which can be complicated. So that's the other thing we thought about it. We'd like to push the system a bit to see if we could do that.
Great, thanks.
I have a couple of questions if nobody else does. One is HVC only used for communication, or does it do other things as well?
Yeah, it's a great question. I think it's most studied for communication in song. There is a little bit of evidence, particularly in females that don't sing. They have a little bitty reduced HVC that maybe it's used in perception of song, and that could also be in these birds that in addition, because it receives auditory input, maybe it's also involved in perceiving song. Beyond that, I don't think there's a lot known.
So what's your thought about the fact that the birds are able to receive effective auditory input when they're anesthetized, if when they're not anesthetized, it doesn't get through? What's its function?
Well, I think what's happening is that I think that auditory input gets modulated. This has been shown in birds in other species, that when they're anesthetized, you see an auditory response, but that is modulated or gated when they're awake. And so it's there, but somehow gated. And so the idea would be that you see that auditory response coming in when they're anesthetized, it's there, but then when they're awake, that gets modulated and changes the circuitry somehow. I don't know. I'd love to hear some ideas. So that now that input is inhibitory.
Is it like that when they're asleep? Is that equivalent to anesthetized or no?
Theoretically, yes, but no. So we did try this. We don't really know if they continue to be asleep, and how you tell if a bird is asleep is not really clear. So we played back song to the bird when they were asleep, and we did not see a response to playback. In other species, sometimes you do see a response. So it definitely depends on the bird species.
If you go to the chat, Kira has typed in several questions.
Oh, thank you. Sorry, I wasn't looking at the chat. I apologize. Okay, let's see. All right, is volume timing, here, should I share my screen? I'm not sure. Okay, now I've lost the chat. Okay, oh, there's all kinds of questions. All right, is volume timing or both sender important for determining the response? I don't know about volume. Here, that's a great question. We haven't tried that. It's hard to change the volume. So I don't know that. I think it is because the male goes where he has very, he's not very loud. So I think that's interesting. Timing, I'm sure it is. They have this code that we don't really understand. They're much smarter than we are, and so they are paying attention to things that we haven't figured out yet. So I think timing is important for sure. Understanding that it's going to be difficult. One of the interesting things these birds do is, in addition to singing duets, they also sing choruses. So you'll have families all singing together. So you'll have the groups of males and females coordinating that activity. And so, you know, clearly timing is important for that information as well. Let's see. Okay, and then we have, if post-inhibitory rebound is part of the self-response activity, should the neural response to their own song be altered in playback? Let's see. I hadn't thought of that. It should be. So do you think that would be, I have to think about that a little bit. It's a great question. But the other part is, the problem with that, when they're anesthetized with something that's blocking that inhibition. So we're not going to get that altered response to playback when they're anesthetized. So that's kind of hard to address. You have a quieter song when they're further away. Doesn't cause it seem less. That's right. So that's the thing that. So the question was, if you have a quieter song when they're further away, but you see longer pauses between syllables, so the quieter song doesn't seem to be causing less inhibition. Yeah, that's right. Which is interesting, and I don't have a good. It's a great question. That's why it's good to talk to you all for these ideas. Yeah, maybe it's just a signal. I don't know if volume matters, at least for the duet. Maybe it may be. So there's some evidence from other birds that these lower volumes, at least when the male is producing the lower volume, is sending a different kind of signal. It's more of like, hey, are you here signal, instead of actually generating a duet. So, and there's probably some other, you know, cues that we're not really paying attention to. I hope that answers a little bit of your question.
Melissa?
Yes.
What's the purpose of the auditory response? If it's, you know, uncovered when they're anesthetized, there's got to be a reason for it. But if it's leading to HVC activity, that's premotor. So yeah, why do you think it's there?
I have no idea. That's a great question. Maybe you know, I don't know. Maybe it's left over from something. Yeah, you know, because this is, it's interesting that if you look at that, it seems that historically, evolutionarily, that both males and female birds sang. And it seems like maybe over history that the females have lost the ability to sing. And so maybe it's kind of a weird leftover from that. I'm not sure. It's a great question. I have to think about it a little bit more.
Is there any, I don't know, I think of it like throat clearing, like if they hear the song and they get an excitatory input and they kind of warm up their vocal cords or something, and then there's a modulation that switches it, so now the auditory signals become inhibitory. Is there anything like that in their behavior?
Yeah, it's interesting. We haven't seen that in HVC ourselves. I have heard it in another bird that only the male sings. I was listening to someone recording when I was a postdoc, and he had an electrode in HVC of this bird, and you heard this background noise coming in that was, and then a little bit later the male sang. So it was this like revving up thing that the bird was doing. And with these, usually they don't just start duetting, but usually one of them will sing solo song, and then the other one will join in. And we think it's actually the female that's kind of like, okay, I'll duet. And so maybe there's a little bit of part of that. It's a gating response to hearing then, and then when you're in the duet, then you're kind of performing a little bit more and helping you. Oh, I like that idea. Yeah, I like that a lot. Yeah, we don't, unfortunately, we don't have evidence for that, but maybe our electrodes are not correct. We're not in the right place. It's coming from somewhere else. I'm not sure.
Any additional questions, comments? Well, Melissa, looks like you're all done. Thank you for the presentation.